Please confirm if my voice is loud and clear. I hope you all can see the screen. So I hope you all are doing good. So everyone, I'm Dr. Tanushree and I welcome you all to another session of Brahmastra Series. So today we will be revising the concepts from Unit 8 and practicing the questions from Unit 8. So countdown has begun, very few days left for the exam. I hope you all are revising well, practicing as many questions as possible. So utilize this 1 hour where we will be revising the concepts from Unit 8, that is inheritance biology, or I should say genetics, which is a very important unit from exam point of view. Very scoring and very important unit. Like 25 marks you can score from here if you answer the part B and part C questions correctly. So in the regular classes, all the concepts have been explained in detail. So if you have missed any point or if you have not revised it yet, then utilize this 1 hour and learn the strategy, learn the trick to solve the questions based on genetics. So let's get started. Here comes the first question on your screen. A series of cell lines was created by fusing mouse and human somatic cells. In a mouse-human somatic cell hybrids, human chromosomes tend to get lost before becoming a stable cell line. Some hybrid cell lines may carry human chromosome deletions. Each cell line was examined for the presence of chromosomes and for the production of an enzyme. The following results were obtained. So here a table is given and you have to find out that which segment of the chromosome has the gene encoding for the enzyme. So in the table, you can see cell line A to E is given, then gene products are given, and chromosomal segments 1p to 5q is given. So it's a part C question. And many students, many CSIR NET aspirants, they think that usually all the questions in part C are very difficult, but no, that is not the case. Even from genetics, there are some very straightforward questions which can be asked in part C. And this is one such example. So those who have prepared genetics thoroughly, they know that this question is from physical chromosome mapping. So answer this question. I hope you all are sitting with a notebook and a pen. So put a timer and solve the question and see how much time you have taken to solve this question. So you have to work on both speed and accuracy. It's a very easy question. Just recall the concept. So to plot a physical map, like there are two maps you all have studied in genetics, right? Linkage map and physical map. So linkage map or genetic map, you can call it as. So just as linkage maps, like they are based on recombination frequency, they are being constructed based on recombination frequency. Physical maps can be constructed on basis of deletion mapping or somatic cell hybridization. So yes, what is the answer here? Try it, solve it. Let me know the answer in the chat. What is somatic cell hybridization? You can see a term here, mouse human somatic cell hybrid. So let's recall this concept first. So here this figure shows the somatic cell hybridization, and basically this technique is used to determine which chromosome contains a gene of interest. So here in this technique, cells from two different cell lines can be fused by treating them with polyethylene glycol or other agents that can alter their plasma membrane. So here you can see that we have taken human fibroblast and mouse tumor cells, and they both are both these two type of cells, they are fused by treating with polyethylene glycol. So in presence of polyethylene glycol, the plasma membrane alters and results into fusion of these cells. So after fusion, you can see the cell possess two nuclei, and we call it as heterokaryon. And eventually, like two nuclei of heterokaryon, they eventually fuse, generating a hybrid cell. So initially, you can see in heterokaryon, the nuclei of human fibroblast cell and mouse tumor cell is separate. But eventually, they'll fuse, generating a hybrid cell, which contain chromosome from both the cell line, that is human fibroblast also and mouse tumor also. Now, the hybrid cells, they divide. So as they divide, they tend to lose chromosomes for the reasons that are not understood. And it is observed that the chromosome from one of the species is one of the species are lost preferentially. So that species is human. So from the cells, hybrid cells, few human chromosomes are lost. And this loss of chromosome, human chromosome, is random, completely random. So human chromosomes are randomly lost from the nucleus during cell division. So not all, but few chromosomes are lost. And mouse chromosomes are retained. So here you can see like these cells when they are cultured, so we get different cell lines. So the difference in these cell lines is that different human chromosomes are lost in different cell lines. Like here, if we have human chromosome 1 or 2, 3, 4, or let's say 1, 2, 3, and 4 are lost from A, then maybe from B it is 2 lost or 6 lost. From C might be 8 or 11 lost, and so on. Right? It's not that all the cell lines will have the same chromosome number being um, lost. So we can use this technique to determine which gene is located on which chromosome. Like basically physical maps, they are being, why do we need a genetic map or physical map in genetics? To determine the position of a gene on a chromosome, right? Their arrangement of the gene on the chromosome. So here we can look for the, like, how will you determine whether a gene is lost? By looking for the product it makes. So human gene can be detected either by looking for protein that it produces, or you can directly look for the gene itself by using hybridization technique, using molecular probes. So that comes under hybridization technique. But here if you are like the question that is based on is detecting a gene on chromosome by looking for its product. So correlation of the presence of the gene with the presence of specific chromosome allow the gene to be assigned to correct chromosome. Like for example, if you see these cell lines, cell line 1, 2, and 3, you will find in cell line 1, chromosome number 2 and 11 are common, right? Now, if we look for the gene product, that is protein or let's say an enzyme, that is present only in cell line 1 but not in 2. So what is different in cell line 1 and 2? Only chromosome number 4. And we are seeing the gene product here, but not in cell line 2. So this indicates that gene is present on chromosome number 4. So we can correlate the presence of the gene with the presence of specific chromosome. And we can even assign the gene to a correct chromosome. Even you can determine like where the gene is located on chromosome. So like here, if you compare cell line 1 and cell line 3, you will find that this portion, like chromosome 4, this portion is deleted. So if the gene would have been here, it will also be deleted. And if the gene is deleted, the product is missing from here. So in cell line 1 and cell line 3, both contain the chromosome number 2, 4 and 11. But here in cell line 1, we see the gene product, but not in 3. The reason being that the region which is deleted has the gene located. So if the gene product is present in the cell line with an intact chromosome but missing from cell line with chromosome deletion, the gene for the product must be located in the deletion region. This we can conclude here. So to answer this question, you have to recall this concept. Okay. So here this table is given to us in the question. So cell line A to E is given. And you can see that plus indicate presence of gene product and minus indicate absence of gene product. And chromosomal segments are also given. So what you have to do is you have to look for the only chromosome common to given cell line, because if the cell line has the gene product, it indicates that the gene is present on the chromosome which are found in these cell lines. So for example, if you see 1p, the gene product is present, like you can see gene product is present in A cell line, B cell line and D cell line, but absent in C and E. Now in 1p chromosome segment, you will see that here like A cell line has this 1p, but not here. So if the chromosome is not there, if this 1p is not there, how come we are seeing a gene product? So that means definitely gene is not located here, right? So the trick here is, instead of going through each chromosome segment or chromosome number if given in the question one by one, just match this pattern given here of the gene product with the pattern given for chromosome segments. So if you look at the plus sign and minus sign which indicate the presence and absence of gene product, you will find it exactly matches with that of 5p. So I told you have to look for the only chromosome common to all given cell line. So 5, it is just showing that it is present in A, B, absent in C, present in D, absent in E. And if it is present, gene product, if we are seeing, that means the gene is located on that chromosome. So it exactly matches with 5p, and therefore answer here is 5p. Beautiful colors, it cannot be 5q. You can see the pattern in 5q is plus for A, plus for B, but for C also it is showing plus. But actually the gene product is missing from C cell line. So if gene product is missing, it means gene is not present here, right? So if the gene would have been on if it would have been on 5q, we would have seen a gene product here, right? So answer here is 5p. Now let's solve another question. So well tried, beautiful colors, but let's solve some more questions. And I really appreciate if all those who are watching, who are live right now, attending this session, please respond in chats, okay? It will give you confidence. When you answer the questions, it will give you confidence. Even if you don't answer it correctly, that doesn't matter. You are here to learn. And whatever mistake you are making, like it's better to commit a mistake rather than not trying at all, right? So please solve another question. So question is given below are statements on concepts of genetics. So A statement is degree to which a particular gene is expressed in phenotype is called. Then B, a heritable change in gene expression that does not result from change in the nucleotide sequence of the genome is called dash change. C, frequency with which a dominant or homozygous recessive gene is phenotypically expressed within a population is called. And an allele that results in death of organisms that is homozygous for the allele is. So based on the given options, you have to find out which of the following options represents the most appropriate sequence of terms to fill all the blank spaces in above statements. And in the options, the terms given are expressivity, penetrance, dominant allele or dominant lethal or recessive lethal or epigenetic or mutation, conditional lethal. So again, it's a very direct question, memory based question. All you need to do is recall the concept here, right? Like a degree to which a particular gene is expressed in phenotype is called what? So in the regular classes, I tell you the keywords, right? I tell you some keywords to learn, which can help you recall the concept faster. Be it codominance, be it incomplete dominance, be it penetrance, be it expressivity. For every term, I have given you a keyword, right? Epistasis. So what do you think is the answer here? So definitely you should know what is expressivity, what is the meaning of epigenetic, right? What is dominant lethal allele, recessive lethal, okay? Okay. Nagasri, you are saying either 3 or 1. Beautiful colors, option 1. Mohammad Saddam Hussain, you're saying option 1. Okay. Well done. Really appreciate you all for trying. So let's see if our answer is correct or not. So first of all, what is epigenetics? Epigenetics refers to heritable change in gene expression that does not result from change in nucleotide sequence of the genome. See, when you talk about mutation, be it point mutation, insertion, or deletion, it causes change in the nucleotide sequence, right? Like if it's a point mutation, let's say G gets replaced with T, or deletion. So deletion will be, let's say GC gets deleted, or insertion where let's say some sequence is being added, right? These nucleotides are added, so they all result in change in reading frame, and these are genetic changes which cause changes in the gene sequence, nucleotide sequence of the genome. But in epigenetics, there is no change in nucleotide sequence of the genome. So how come if there is no change in the nucleotide sequence of the genome, how it can how it can change the gene expression? So the examples of epigenetic changes are like methylation. Let's say a methyl group is present over here. Okay, methyl group attaches over here. Now maybe this is a region where the transcription factors need to bind. How a gene is expressed? We all have read about central dogma, right? So if the transcriptional factors at this place, if methyl group is attached, the transcriptional factors will not able to bind here, right? So gene will not be able to express. It will not be transcribed into messenger RNA, which in turn eventually will not be translated into protein, right? So epigenetic changes, they doesn't result from change in nucleotide sequence. They are just occur because of the modification of certain regions of the DNA which affect the gene expression. So example is methylation. Then next term, another term given was penetrance. So if you remember, I have given a keyword in the class, P for penetrance, P for phenotype. It is related to phenotype. So penetrance is the percentage of individuals, and it is always expressed in percentage, that show a particular phenotype among those capable of showing it is called penetrance of the genotype. So in the class, we all have discussed this very popular example of the Hawaiian bird, blue egg color bird, like blue egg bird when crossed with white egg bird. So where the blue being, the allele for blue color being dominant over white. So the F1 generation will be heterozygous, right? So we have considered all the cases. If there would have been a complete penetrance, so let's say 9 eggs, so all are heterozygous. So what color we should expect? Blue color. So if all are blue, okay, so that means if we get the like if the individual show a particular phenotype, it is capable of showing. Capable of showing means it has a genotype for it. So when the genotype is heterozygous, we expect the blue color eggs, right? So if we get all the blue color eggs, it means complete penetrance is there. But like here in this example, out of 9, you can see only 5 are blue, rest 4 are white. So although these which are white, they are not homozygous recessive, they are heterozygous only. They should have shown blue egg color. So this is a case of incomplete penetrance or variable penetrance. And third case is when all are blue, but you can see they are not of exact same shade. Like this is very dark, this is like intermediate color, this is light, and like this is very like the color goes so light that it appears white. That is expressivity, variable expressivity. So expressivity is related to expression, degree of expression. So degree to which a penetrant gene or genotype is phenotypically expressed in an individual, that is expressivity. So here we don't see the same degree of expression, same blue color, intensity of blue color, right? So it is variable expressivity. And what is lethal allele? A lethal allele is which results in death of an organism. So dominant lethal allele is which can show its effect in both homozygous and heterozygous condition. But if it is a recessive lethal allele, then it has to be in homozygous condition, right? So lethal allele is which causes the death of an organism. So in the question, like if we go through the terms, the degree to which a particular gene is expressed in phenotype, we call it as expressivity, right? Then heritable change in gene expression. So when I say expressivity, A is expressivity, it is only given in option 1, not in 2, not in 3, not in 4. Then heritable change in gene expression that does not result from change in nucleotide sequence of the genome, that is epigenetic. Then frequency with which a dominant or homozygous recessive gene is phenotypically expressed within a population. So frequency, that is penetrance. An allele that results in death of an organism, that is homozygous for allele, that is recessive lethal. So here the answer is option 1. So very well done, Divya, Mohammed, Saddam Hussain, Nagsri. Beautiful colors. You all have answered correctly. Very good. Keep it up. Moving over to third question. Epistasis is observed between different genes in flower color of sweet pea. A red variety on selfing yields red to white in ratio 9:7. The precursors and intermediate give white color. The following biochemical pathways were proposed for the above observations. Which one of the options represents the correct pathways that explains the observations? Now again, sometimes what happens is, it happens with most of the CSIR NET aspirants that as soon as they look at the question and they see the pathway or they see any flow chart, they get scared, or any figure, they get scared that okay, this question is very lengthy, or this question is, it must be very difficult. But remember, it's not always the case. As I told you in the beginning, that part C questions can be sometimes very easy, very direct. Like here, all you need to do is just pick the hints given in the question. And the best part of such figure-based question is that you don't have to think any pathway-based or figure-based question is you don't have to think out of the box, right? So here you can see a biochemical pathway is given. So here you all need to recall about epistasis. What is epistasis? Then in what kind of epistasis you get this ratio 9:7. So just recall about that. So yes, tell me the answer for this question. In what kind of epistasis you get 9:7 ratio? Yes, let me know in the chats. Just look at the pathway A, B, and C. So this question is from subunit C, extensions of Mendelian principles. So in the class, I have told you to learn the examples, right? To learn the ratio. Few tricks I shared in the class, right? That how you have to learn about different type of epistasis. I can totally understand that learning about different kind of epistasis can be quite confusing, right? So I've shared a trick in the class. So just recall that. Focus on the ratio, focus on the example given, flower color of sweet pea. So let's recall first what is epistasis. Very good, Divya. Yes, you have answered it correctly. Duplicate recessive epistasis. Well done. So what do you think is the answer here? Which option represents the correct pathway or pathways that explain the observations? Only A, or both A and B, or both B and C, or all three, A, B, and C? So I hope everyone is solving. So what is epistasis? Epistasis is a type of gene expression where one gene masks the expression of another gene at a distance locus. That is, we consider non-allelic case here, right? That is, we consider alleles of two different genes. So let's say we have one gene which has a dominant allele, this one, and recessive allele, this one. And this is another allele with so this is the dominant allele of the gene and this is the recessive allele of the gene. So the C and P, they are non-allelic, right? So epistasis is a kind of gene expression where, like when we talk about gene interaction, so one gene masks the expression of another gene. So it might be a dominant allele of a gene which can mask the expression of either allele of a gene at a distance locus or vice versa, right? Or it can be a recessive allele masking the expression of either allele of the another gene, right? So in the question, the example given is flower color of P, which is an example for duplicate recessive epistasis. And this 9:7 ratio we get in duplicate recessive epistasis only. So from the question, you can easily figure out that, okay, this question is about which kind of epistasis. But since pathways are given, so here you have to recall the concept that why this 9:7 ratio is achieved. So if we consider a hypothetical example where we consider that we have a precursor which is white, and then we have an intermediate which is also white, but the final product is colored. Okay. So you can see that in this pathway, in this entire pathway, the product of two genes are involved. right? So here we need a C gene product, that is enzyme C, which convert precursor to intermediate. And then conversion of intermediate to product, we need a product of P gene, that is enzyme P. So that means we need this condition where at least you should have one dominant allele of C gene and P gene. Okay, so dash here means either homozygous dominant condition or heterozygous. Same for P, like either homozygous dominant or heterozygous. So at least two dominant allele is needed. But what if the C gets mutated? Let's say the C gets mutated, C gene gets mutated, and you have the recessive allele C, which is or mutant allele. So pathway will be blocked here. It doesn't matter you have the dominant allele P, because at the end, the product of gene P can act only on intermediate. It cannot act on the precursor. And intermediate can only form when precursor is converted by the product of C gene, dominant C gene, C allele. So that means when you have this condition where C allele is mutated, you cannot get, this pathway cannot be completed. Similarly, if you have the dominant C allele, but P gets mutated. So in this case also, the pathway, like the precursor will be converted to intermediate, but pathway will be blocked here, right? So you have to consider both the cases. So one case I told you, another case is where you have the dominant C allele, but the P gene gets mutated. So pathway, like precursor will be converted to intermediate, but pathway here will be blocked. So in this case also, you will not get the purple pigment, right? And if both gets mutated, then definitely no pathway will be completed again. So this case we have seen in the, like this thing we have seen in the example of flower color of P, where when you cross the one white variety with another, you get all the progeny purple. So all progeny purple will only be possible when you have both dominant C and P. Like if you see the plant parent generation, here you have dominant C, but homozygous recessive P. Here it is homozygous recessive C, but homozygous dominant P. So one of the dominant allele is absent in both the plant varieties. But in F1 progeny, since both have contributed the dominant allele, the pathway is completed. So wherever both the dominant alleles are coming together, right, you can call it as, like, you will get a purple pigment. And wherever it is missing, you can call it as, you can write it as, like, the pathway will not be completed, so white. So in terms of epistasis, how will you understand? In terms of epistasis, like if I just show you the genotype, you will say that, ma'am, because dominancy allele is present, so we should get a phenotype of dominant allele. But we are getting white, right? We are getting white. So, like, even if you see here, if I just show you the genotype, if I don't show you the color, you will say that, ma'am, we should get a phenotype of dominant allele, which is purple. But we are getting white. So in terms of epistasis, we can say that the recessive allele is epistatic to the dominant allele of the gene. So that is why it is called as duplicate recessive epistasis, because the recessive allele of both the genes, like if you take P or if you take C, they both are epistatic to the dominant allele of another gene. So the color, you can say that, like here, the ratio we will get is 9:7. So only 9. So Mendel's dihybrid cross phenotypic ratio 9:3:3:1 here is modified to 9:7 because of this kind of epistasis happening. Now, since 3 pathways are given to us, A, B, and C here, right? 3 pathways are given. So here you can see that in A pathway, precursor is converted by product of gene A into intermediate X, and this intermediate is converted to the final product, which is read by product of gene B. In B pathway, we have a precursor which is converted to precursor intracellular by product of gene A, and this is converted to final product by product of gene B. So if A gets mutated or B gets mutated, then in both the cases, the pathway will not be completed. And if you see the C pathway, it is A or B condition. So if this would have been A or B condition, we would have not got 9:7 ratio, right? We would have got red in that case. We would have not got white if A or B condition is there, right? Here it should be, we need both A and B, right? So remember, duplicate recessive epistasis, it is also called as complementary gene interaction. And here the gene product of dominant allele of both the genes are required for color formation. So both pathway A and B can explain the observation, and therefore answer is option 2. So Nagasri, Divya, you have answered correctly. Divya, I'm glad like at least out of 4 options, you are able to omit 2 options out of 4. So well done. Now just work on omitting 2, like you are left with 2 options. So now try on try on answering accurately 1 option, okay, out of 2. So everybody kindly note, we are coming up with a data science for biologists. So that is 120 days hand-on internship. So you will be like, you will get a chance to work on a project and paper publication, and also work experience. So it is starting 4th March. Okay, so the projects will be both online and offline. And also we are like, don't forget, please note the date. On 1st March, there is a big data biology national workshop. So please, you can note the dates, and for details, you can call on our toll-free number, which you can see on your screen. So yes, are you able to solve the questions? Can I get some thumbs up? So I hope these sessions are really helpful when you are solving the questions, learning the strategies and getting the answers. Okay, that's great. Thank you so much. So moving over to another question. The following represents selected AFLP bands 1 to 5th observed in parents P1 and P2. So this is parent P1, this is parent 2, this is F1 is the progeny. And 20 double haploid progeny developed from F1. Now, double haploids are created through chromosome doubling of pollen grains in anther culture. The following statements were made about the above AFLP bands. A, band 1 and 4th are allelic. B, band 2 and 5th assort independently. And C, band 3 is uninformative. Which one of the following options represents a combination of all correct statements? A only, C only, A and B only, or is it A, B, and C? So here you can see this question is a combination based question. Like usually in unit 8 also, or like you can say it's based on molecular markers, not a combination based. Actually, unit 13 also you study about molecular markers, right? And in subunit D of unit 8 also you study about molecular markers. So it's all about the molecular marker AFLP. So I hope you all know the full form of AFLP, amplified fragment length polymorphism, right? So here are some statements given. So you don't have to think out of the box. Again, like you just go through this profile, gel profile, which is given, see the band pattern, and based on that answer the question, whether the bands are allelic or assorting independently or they are uninformative. So double haploids created through chromosome doubling of pollen grains in anther culture. Okay, Sarita, 1 and 4 are allelic. So you're saying it is true, right? So you have to choose a combination of all correct statement. Options represents a combination of all correct statements. So if you are saying 1 and 4 are allelic is the answer, so that means you're saying option 1A only, right? Yes, Divya, Nagashri, Mohammed Saddam Hussain, quickly answer the question. And others who are solving, please write in chat. See, how will you go? How will you read this gel profile? It's like P1, you can see some bands, right? Like in P1, the band is over here. In P2, it is not in over here. For 3, you can see it is showing all like bands and all for P1, P2, F1 and all the double haploids. Then you see some change in 4 and 5th also. So to answer this question, we will consider each statement one by one. Okay. So Nagasri, you're saying option 4. Let's check. Let's check now. So basically, as we all know, when the gametes are formed, alleles they segregate. Okay, according to Mendel's law, according to Mendel principles, we have studied about principle of segregation, law of segregation, and we know that alleles they segregate, right? So, like an individual, it is diploid, but when the gametes are formed, gametes are always haploid, so alleles they segregate. So each gamete receive an allele, right? So that is why we call it as a law of purity of gametes. Also, law of segregation is called as law of purity of gametes. So the first statement is band 1 and 4 are allelic. So what pattern do we see here? This is 1 and this is 4, right? So bands 1 and 4 are allelic. So you can see that wherever the bands present, like in 1, wherever bands are present, they are not present in 4. Like you can see these are the bands over here, but these are not present over here, right? And in 4, the bands are present over here, but these are not present over here. So yes, this indicates the statement A is correct. Because obviously when the gametes are formed, the alleles segregate from each other, go to separate gametes. So gamete will have only one allele of a particular AFLP. So this statement is correct. Then second is band 2 and 5th assort independently. We all know very well that to know, like through test cross, we can determine whether it's a case of linkage or independent assortment. So we know in case of independent assortment, we get equal proportion of recombinants and like parentals, right? So here P1 and P2 are parent and F1 is the progeny, right? Now we have to look at bands 2 and 5th, right? So how can you say they are assorting independently? So basically, you will see that in double haploid progeny, there are equal numbers having only 5 and only 2. Like if you see here, these are 5, which are common in both, right? And you will find that these are only present in 2, 5, and these 5 are present only in 5th. And rest 5, you can see they are missing in both. That is, they are neither in 5th nor in 2. So here, The number of parental is number of recombinant. That means they are assorting independently. So statement B is also correct. So when I say A and B is also correct. So answer cannot be option 1, cannot be option 2. It will be between 3 and 4, right? So third is band 3 is uninformative. So actually, if you see here, the third one, it is like all for all we are getting the bands. We call it as monomorphic. So basically, monomorphic bands are those which are common in all, right? And they don't tell us about the genetic variations. It's the polymorphic bands which tells us about the variations. So yes, it is not informative. They provide no information about the polymorphism. So C is also correct. And hence the answer here is option 4. So well done, Nagashri. Well done, Sarita. At least you are able to omit 2 options out of 4. So here answer will not be 3, it will be 4. And then coming to this question, in Drosophila, a cross was set between a male homozygous for allele and a female homozygous of. So this is the homozygous for this allele. Okay. So you can see this is the homozygous dominant condition. This is homozygous recessive. But just focus on the phenotype. When this is the genotype, the phenotype observed is A type. And when this is the genotype, homozygous recessive, the phenotype observed is B. And even it is mentioned in the question that S plus being a dominant allele and only S is a recessive allele. Now, F1 individuals, they had phenotype B. And when F1 individuals were crossed among themselves, all progeny obtained were of phenotype A in F2. So following explanations were proposed for the results obtained. Is this an example of cytoplasmic inheritance? This is exhibiting genetic maternal effect. This is a quantitative trait influenced by the environment. This is the gene interaction with epistasis. This is exhibiting gene interaction with epistasis. Or the trait is showing position effect variation. Which one of the following option is correct? Now see, instead of getting confused with the terms given, if you simply focus on this experiment or this example given of the cross, you can easily infer the answer even without looking at the explanation. Like just focus on this example and recall the term and look for which like A to E that term is given. And based on that, you can find out which option is correct. So this is the last question for today, because at 7 we will be coming live. We are having a webinar, so you all can attend. So quickly answer this question. So note it down, okay? Read the question carefully. What all information is given to you in the question? The answer is in the question itself. All you need to do here is just recall where you have, like, where this concept is applicable. Is it a maternal effect? Is it a quantitative trait? Example, it's a epistasis, it is cytoplasmic inheritance. So I hope you know cytoplasmic inheritance, where we talk about inheritance of mitochondria, chloroplast genes, right? Yes, Nagasri, Sarita, Mohammad Saddam Hussain. All those who are watching, please answer this question. Analyze your preparation. Check your performance. Okay, Divya, you have mentioned maternal effect. Sarita, maternal effect. Okay, option 2. Good. Nagasri, for this question, what is your answer? So please prepare unit 8, okay? It's very scoring and important unit. In the beginning only I told you. Every year around 3 to 4 questions, part B and part C are asked from this unit. So 30, like 25 marks you can easily get from here. At least 25. Or like 22 to 25, you can keep it like that. Last 1 minute. Quickly answer this question. Focus on the information given in the question. And those who are attending live, you can see a link on your screen. In the chat, you can see the screen. You can see the link. So you can download some more CSIR NET Part C questions for practice. So this is only for those students who are attending live. So make sure that you go to that link and get access to more CSIR NET Part C questions. So coming back to the question, Nagasri, you are saying option 2. Fine. So let's check. So basically, I told you write what all information is given in the question. So the male is homozygous dominant. So it is already given that this allele, S plus allele, is a dominant over S. It is recessive. So homozygous dominant male showing phenotype A is crossed with female, homozygous recessive with phenotype B. So when they are crossed, the F1 progeny was all obviously heterozygous when we are performing this type of cross. So obviously the gametes form will be with the dominant allele here. This parent will form the gamete with the recessive allele. And when they are fused together, so F1 generation will be heterozygous. But you can see the phenotype is observed is of B. So B phenotype is shown by the female, the recessive, homozygous recessive condition, right? And further, when F1 was selfed, when selfing of F1 generation was done, the F2 progeny showed phenotype A, right? So as you know, when selfing of F1 generation is done, the both parent, male and female, will be heterozygous. And phenotype A we are getting. So obviously this is a case of maternal effect, right? Because F1 having phenotype B, having a genotype for phenotype A. But then how can we are getting phenotype B? So through maternal effect, we can explain it. So maternal effect is where the progeny phenotype is determined by the genotype of mother, not the phenotype of its own. So maternal effect in genetics is the phenomena where genotype of mother is expressed in phenotype of the offspring. So phenotype of an individual reflects the genotype of mother rather than genotype of individual. And in F2, all the progeny is showing phenotype A. So this indicates that again, it's the case of maternal effect. Because only 1/4 progeny, which is having homozygous recessive, should show this genotype. But entire F2 has phenotype A. That means it's because of maternal effect. So answer here is option 2, and very well done. You all have answered correctly. So everybody, I'm stopping here because at 7 we are coming up live. Stay tuned. Don't forget to attend the webinar and learn something new, and all the very best for the exam. So please practice as many questions as possible. revise. You can take help of Biotecnika revision tools for quick revision and recall, and take care of your health. That is most important. And the link for practice, more questions you need for practice, the link is on your, like you can see it on the chat. So thank you everyone and take care.